All right. Good morning, Awafria. Welcome to our last day of STEMCon. We are so excited for our speakers right now, specifically our live closing keynote by Dr. Hochstrasser. We wanted to remind you of a few things before we get started. If you have any questions throughout, again, you won't be able to type them into the YouTube Live, but you will be able to use that Mentimeter that is in the same document as where you got the link for the live stream. So all you do is go to menti, menti.com, and the code is 1604027. And that will stay in that document. So if you have anything you want to chat in, any shout outs, any comments, any questions, it all goes in there. And now I would like to welcome one of our STEMCon committee members who has been instrumental in putting this whole event together and is a student at Millennium High School, Miss Bianca Perez.
Dr. Megan Hochstrasser, who works with Dr. Jennifer Doudna, Nobel Prize recipient in chemistry in 2020, will give us an introduction to CRISPR genome editing and how it can be used to learn about nature and cure disease. She will also discuss ethical questions that arise with this newfound capability to alter ourselves and our world. Megan Hochstrasser has a BA in biology from Brown University and received her PhD from Jennifer Doudna's lab at UC Berkeley in 2016, where she studied mechanisms of CRISPR immunity in bacteria. She joined the Innovative Genomics Institute in September of 2016 to handle communications, hoping to bridge the gap between researchers and the lay public. In spring of 2020, she shifted to leading IGI's education efforts full-time.
All right, thanks so much for that introduction. I'm happy to be here with you all today. Let me just share my screen. Oops. Okay. Great, so hopefully everyone can see this. All right. So thanks again for having me. Great to be here. As was introduced very nicely, my name is Megan Hochstrasser, and I work for a group called the Innovative Genomics Institute. And I am a specialist in this thing called CRISPR. So this is a mechanism for rewriting DNA. And so with that potential, we can also rewrite the future of our planet and humanity. Okay, so just briefly, the IGI, or Innovative Genomics Institute, is a place where we're trying to make sure that these new genetic engineering tools that we're developing are used for good reasons and the benefits are really maximized and applied equitably to people. So we're a partnership between UC Berkeley and UC San Francisco. So we're, a nonprofit research group. We're not a company, and we get to fund all kinds of different cool research around the San Francisco Bay Area. My role at the IGI is focused on education and outreach. I kind of think of it as science communication in general, like helping people understand our science through talks like this, through apps and games that we develop, through online resources, all kinds of things. So it's a really fun position that I feel very fortunate to have. And I'm going to be telling you a little bit about how I got here, and then the rest of the talk will just be about CRISPR. So who am I? As was mentioned, I got my PhD studying CRISPR at UC Berkeley, where I ended up joining Jennifer Doudna's lab. And this was an incredibly lucky thing that I ended up doing because it was kind of right before this whole CRISPR craze, and I just was in the right place at the right time and got to witness the development of a really important technology. So I've worked in a lot of labs over the years in undergrad, getting my hands dirty in the lab, trying to figure out if I like doing research. And I've also worked in various labs during the initial part of my PhD. And they're all really cool in different ways. Like, you don't have to join a lab that becomes kind of famous in the science world to be successful. But I was really lucky to be in this lab. So it was a great place to be. And my boss at the time, Jennifer, she had sort of stumbled into this technology that has a huge power to change our species' genetic future, as she says. And so she got really involved in the ethical discussion around how to use this tool, in addition to kind of being a pioneer in the science. And so while she was, kind of grappling with these ethical questions and hoping that this discovery would take off and have a good impact on the world, she decided to create the Innovative Genomics Institute. So she wanted to make an institute that would really push the science and be at the forefront, but then also lead this sort of moral discussion. So around the time that I graduated, she started the IGI. And so I started working in IGI in communications. And I don't have time to get into like my career path that much. So I'll just link you to 2 resources that you can check out if you really are interested in a potentially science communication career that might seem a long way off. And usually you kind of have to just go towards a generic science path first. But I think I give you some tips and kind of share what my path was in these 2 resources if you're interested. Okay. So that's it about me. I'd be happy to answer questions about that later at the end. But for now, I want to get into genetic engineering and CRISPR in particular. So what I'm showing you here is an artist's depiction of DNA. So DNA is like the central thing that I'm thinking about all the time. It's the blueprint of life. So all of our cells, all of the cells in flowers and cows and everything that is alive have DNA. So that's like the instruction manual for helping cells and organisms grow, reproduce, etc. And what DNA does is control traits. So this is I take this from 23andMe, and please don't think it's an endorsement of 23andMe, but these are sort of some of the traits that you can test yourself for if you do one of these like personal genetics kits like 23andMe. So they'll tell you how your unique genome is tied to various traits, like whether or not you have dimples, whether or not you like cilantro, things like that. And so that's a little bit frivolous, sort of, like you probably already know if you have dimples or freckles. But there's also a lot of kind of less fun things that you can learn about in your DNA. So in your genetic code, if you have little errors that have accumulated, or that you have from birth that are passed down from one or both parents, you can actually have a lot of different genetic diseases. So a few of these are listed at the right. Probably the most well-known in this list is cystic fibrosis. And so that's technically a rare disease. It doesn't affect that many people, but it's a very, very serious disease, and there aren't a lot of great treatment options. And these other disorders that are in this list are even rarer than that, but they're still really significant. So there's kind of this problem in the health treatment world where you could have such a rare disease that there are so few other people that have it that, one, it's really isolating as an experience, but 2, it's not really incentivized for drug companies to try to develop an entire drug for like 30 people on the planet that have a disorder. So it's very clear that we've needed better solutions or a solution at all for genetic diseases. And that's why people are so excited about CRISPR. So CRISPR is a tool that lets us change DNA. So as I said, we've known what causes a lot of these genetic diseases for decades and decades, but we haven't been able to actually go into the underlying problem, the DNA, and fix it. We've only been able to kind of manage symptoms in most cases. So CRISPR is a unique tool because it lets us not very easily, but more easily than before, it lets us change DNA. And that's what I'm showing you here, which I won't show you this structure for too long, but this is kind of like an artist's depiction of what this one particular CRISPR protein called Cas9, that's the most famous one, how it looks and how it works. And so I'll explain in a bit how it actually works, but I wanted to show you that, just because I think this is cool, that really is what the protein looks like. Like, that's not just a crazy fanciful thing that an artist came up with. And you can actually this is called atomic force microscopy, to visualize these molecules directly. So this is actually a video of this incredibly small microscopic protein, single protein on a strand of DNA. And there, it just cut the DNA. I think that's so cool. So yeah, it really looks like that. And this is a DNA cutting enzyme. So let me explain how this all works. So Cas9 is basically a protein that you can program to go cut wherever you want in DNA and make an edit at that site. And what's so revolutionary about Cas9 is how easy it is to program it to cut somewhere new. So to do this, the first step is to find your target. So I'm showing you a target site in blue. We could imagine there's a mutation there. Maybe this a cystic fibrosis mutation is at that site. So if we want Cas9 to go there to make the edit, we have to program it in a particular way. So I'm showing you the protein itself in green. That's like the little molecular machine that does the cutting. And you'll see that it's carrying around this squiggly thing. So this is called the guide RNA. And RNA is, you don't have to worry about it too much. It's similar to DNA. It's just kind of more flexible. It can be a single strand. It can fold into these different hairpin shapes. And the guide RNA serves as the location code, or like the GPS for the protein. So part of it, the part that's on the right that's all like coiled up, that's sort of like the handle that Cas9 holds on to, and that never changes. And then the part that's in blue is something that we can change as scientists. So we make it so that that little sequence of RNA, sorry, in the blue matches the sequence of DNA at the target. And that's how we tell Cas9 to go make an edit at that site. So how does it do that? It actually base pairs with the target. So it unwinds those two strands of the target DNA, and its guide RNA ends up forming like Watson-Crick base pair. So, or yeah, like a double helix basically with the target. And so if Cas9 sees like, yes, I could base pair all the way through this, I have a match, then it'll make a cut. And so that's what you see on the right. It cuts through both strands. And so that's the first step of gene editing. And all genome editing starts with making a cut in DNA, whether it's CRISPR or another tool. So why does that happen? It might not seem like cutting DNA would be curing a disease, right? So let's imagine we've made this cut. It's called a double strand break because it goes through both strands of the helix. This sort of sends the cell that we're in into a panic. So cells will not survive if they have their DNA cut up. And so they have all these repair mechanisms in place to kind of troll around looking for breaks and fixing them. So this little break can get fixed in one of two ways. So the most common is that the cell's repair system will kind of just like do a crappy job and slap the two ends together. And it'll sometimes kind of like throw in a few genetic letters or nucleotides. It'll cut a few out. It's really just like this desperate, like, I must close this gap sort of reaction. And that sounds sort of bad, but it can actually be really useful because this can end up knocking out a gene, which means if you think of a gene as like a little instruction manual for a protein, if you ended up like tearing out a bunch of pages or adding random ones into an instruction manual, you probably wouldn't have a functional product when you assembled it. And so this is nice to do when you're doing just basic research. Like if you want to understand what a gene does in this plant that is next to me, I could potentially knock a gene out and then see what happens with the plant. Maybe the plant would turn white, and then I would learn, okay, that gene's maybe involved in like photosynthesis or something to do with chlorophyll or things that make plants green. So that can be really useful. But in the case of something like cystic fibrosis, a genetic disease that we want to cure, you don't want to further mess up a gene. You actually want to fix it. So the way that we could do that is to add in what's called a donor or template DNA, which I'm showing here in purple. And this is like a little piece of DNA that we provide as the scientist that the cell will paste in kind of like a patch to seal that gap. And by adding this in, we can kind of trick the cell into either adding totally new DNA or replacing damaged DNA. So we could, in theory, replace a mutation that's causing disease. So there's these two pathways. I could talk more about how we control which one actually happens and when we can't really control that later, but just know that these are the two ways that a gene editing cut can be resolved. Okay, so I have a little video here to try to kind of summarize what I've just said and show gene editing in action. So we're going to dive into a cell, and we're going specifically into the nucleus where all the DNA is stored. And so Cas9, we've added to this cell. It's sort of searching around through the DNA. You can see these different Cas9s looking around, but not quite finding their target. And then this Cas9 is actually going to find the correct target. So it's going to roll up to the DNA, and it's going to slowly unwind the two strands. You can see they're unwinding, and its guide RNA is actually winding in and base pairing with the target. And then it made a cut through both strands once it found that match. So then the Cas9 is going to get knocked off. One of these repair systems is going to jump into action, And there you can see in pink, we've actually made a change. We've added 2 nucleotides at the end of that. So that's kind of just like a brief little summary of how that works. Again, this is an artist conception. And it's actually, whoops, a really great artist named Janet Iwasa. So I like to point her out because people don't know that you could be like a molecular animator as a career. So she actually makes, as her full-time job, she makes animations and graphics and things like this to try to show how science works, which is super cool, I think. So if you don't get your job at Pixar that you want, you can work for Janet Iwasa. Okay, so why does this matter? What are we trying to do here? We really just want to change DNA. So that was a lot of kind of molecular detail. And I know if some people really want that, some people are like, I have no idea what that was. So I try to kind of stop at this point and summarize and kind of walk through what we're doing with CRISPR. So say we have a gene that has a mutation, and here I'm showing you, a metaphorical sentence. So pretend this represents our gene, and in red, we have a mutation. So it shouldn't say twinkle, twinkle, battle star. We could, in theory, use something like CRISPR to edit that sequence to what we want, or the correct sequence, twinkle, twinkle, little star. And because of this editing analogy, people often talk about this as like, imagine you had a keyboard for DNA, and editing is a good analogy, I think, in some ways, like text editing. The other thing people kind of compare this to is surgery, like a genetic surgery. Imagine using a scalpel and making cuts and removing bad pieces of or harmful pieces of DNA and replacing them. So these different metaphors can work for different people. I think as long as you remember that CRISPR is letting us change DNA, you can understand all the things we can potentially do with it. So why is this so important? Why is this such a big advancement? I'll just talk briefly about this. So there have been prior gene editing tools. So I'm showing you on the left 2 of these, ZF and TAL. So that means zinc finger and TAL nuclease. So these were the prior tools, and they have not been replaced, but CRISPR has sort of become much more dominant, because with those tools, you'll see in orange the part that you need to change every time you want this gene editing tool to go to a new place in DNA. And in zinc fingers and talons, that thing that you have to change is actually protein. And engineering a new protein every time you want to edit somewhere new is really difficult. Like, protein engineering is a whole field in and of itself. So that could take you months and months. But with CRISPR, you'll see in orange, the element that changes is just that piece of RNA that we talked about before. And RNA is much, much easier to change than protein. So I could literally go to a website and order a DNA template to make RNA, or even order the guide RNA itself, and just type in the sequence that I want, just like I'm showing you here, and have like a tube of this delivered to me the next week or the next day if it's DNA. So it's really, really easy to do this, and it's a lot cheaper than the other tools. So that's kind of why this took off so quickly when it was developed in 2012, 2013, is that it was just like really user-friendly and easy to do. So how has this taken off, and why? I think one big thing that I like to mention is the impact that this has had on just doing research. Even if we never get to cure anyone of any disease, it's already transformed what we can do in the lab. So that's because of what I'm telling you here, which is that if something has DNA, we can now edit it. And these handful of organisms that I'm showing you on this slide have one thing in common. These are all so-called model organisms. So if you were in a science lab in 1980 or something, you'd probably be working on one of these, and only one of these. So that means mice, fruit flies, Arabidopsis as a plant model organism, yeast. These are sort of the go-to organisms where we had genetic systems. We were able to sort of modify their DNA in different ways, but it was really specific to those organisms. And now with CRISPR, we've actually been able to edit the DNA of hundreds of organisms with just one tool. So everything I'm showing you here has been edited with CRISPR, and I made this slide like 3 years ago or something. So there's been many, many more things edited since then. And I think this shows you just the types of questions that we can start to ask about the really beautiful diversity of life around us. So nature has done really cool things, and now we can kind of start to understand how that works at the genetic level. Like you'll see there's in the third column, there's a bunch of butterflies. People are now editing butterfly DNA to understand how those wing patterns are formed. and they can actually control and have different wing patterns based on how we edit the genome of the butterfly. So there's just really interesting things like that, or very interesting to me anyway, to know how nature is doing some of the amazing things that it does. Okay, so I think one of the things people are really excited about though with this technology is of course potentially treating disease, right? Basic research is fun, butterflies are cool, but there are people that are suffering that we should be trying to help. So one of the kind of first diseases that's really being investigated as a possible target for CRISPR is sickle cell disease, which you might know as sickle cell anemia. So this is also a rare disease, and it basically, there's a mutation in this hemoglobin gene. So hemoglobin is this protein that is in your red blood cells that helps carry oxygen around. And in people with sickle cell, they have a mutation in that protein that causes it to stick together, and it ends up distorting the cells and taking them from the nice, like round red blood cells that are in most people's in bodies and turning them into this sickled shape. So they get stiff and elongated, and they're kind of like little sticks. And so they end up piling up inside of blood vessels. So you can see here in this again, artist depiction of what this might look like. These cells like get hooked on each other. They can't go smoothly through the vessel. And so they end up clogging. And this means oxygen and blood are, is not getting to extremities, to different organs, to various tissues. And if you don't get oxygen, the tissue is going to die. So this is a really, really painful disease. It's characterized by having these horrible pain crises. And it's also damaging the tissue. And so this damage can accumulate and really reduce the lifespan of people with sickle cell, in addition to it being just a really horrible disease to live with. And so it's actually a good candidate for CRISPR because we have just a single letter that is mutated in this disease, which I think is almost, amazing that you can have just one typo in your genome and have such a profound effect. But that's actually a good thing for CRISPR because it's simple and it's something that we can potentially fix directly. So this is something that we're developing at our institute, trying to fix this directly. But luckily, since we're slow, other people are also doing this, and they're doing it in a slightly different way that I won't get into, but it's also using CRISPR to try to make a genetic change that will treat the disease. And so I like to show this slide because it's like the proof in the pudding that this could actually help people. So this woman's name is Victoria Gray. And she has a really amazing story. I'm really shocked that she's volunteered to, talk about her experience participating in a clinical trial. So she's one of 2 or 3 people in the world, basically at this point, who have undergone this experimental treatment. We need to start with a small number of people to make sure things are safe before expanding. And she has basically become symptom-free of sickle cell disease after getting this treatment. So incredibly profound effect. It's been really interesting to follow her story on NPR. If you want to Google her, NPR sickle cell will give you a lot of articles. So there's a lot of promise here, and there's actually quite a few more clinical trials, less than 10, but too many for me to talk about. And so I recommend, if you are interested in learning more about this and what's being done right now, to check out this blog post that we have on our site from 2019. It's a little bit outdated, but we're actually very soon, hopefully, coming out with an update for 2021 that is like a comprehensive guide of every single trial that's happening right now. Okay, so for the, I think, remainder of the talk, I want to talk about the ethical decisions that this technology forces us to think about. So if you've seen the Spider-Man movie, you'll know that with great power comes great responsibility, right? So if we can do something, that doesn't always mean we should do it. And I try not to, ever vilify a technology itself. Like, I think it's the people that use technology that can choose whether or not to use it for reasons that are good or bad. And so because we now are able to change DNA, we have so much potential to do things that might seem good to one person, might seem controversial to somebody else. So a few examples would be like genetically modified foods. I think they get a really bad rap for a mix of good and bad reasons. But in theory, scientifically, my scientific perspective would be that there is no reason to think that modifying a food is going to make it harmful. I think there's a lot of things we can do with our crops to improve them, making, drought-resistant rice or flood-tolerant rice, or different kinds of crops that could potentially withstand the effects of climate change. You could potentially make more nutritious crops or remove allergens, like could we make non-allergenic peanuts, for example? I think there's a lot of really cool things we could potentially do. And CRISPR is a huge advancement over the prior tools that were used to make GMOs. So we have a lot more precision now. And so even though there are these like social conflicts sometimes, I think there are also big potentials for good in that space. But it's the kind of thing we have to talk about and decide what we want as a society. I'm also just putting a little mosquito here to note there's this whole thing that I can't even begin to explain called a gene drive that could potentially mean that we would use CRISPR to wipe out a population of mosquitoes. Like what if we could wipe out the species of mosquito that carries malaria, for example? That sounds kind of wild, right? And could have really terrible ecological consequences to just not have a species anymore. But on the other hand, maybe it doesn't have those consequences. The ecosystem rebounds and then people don't get malaria anymore. And there's so many people, there's such a profound, horrible effect of malaria, especially in the African continent. And it kills hundreds of thousands of people. So there's these huge moral questions, like, do you try to do something that could be ecologically devastating because it could have such an amazing impact on people's health? Or is that not the right solution? Should we be spreading mosquito nets around instead? Lots to think about. And then last, I'll just get into kind of the most frequently discussed CRISPR ethics topic, which is the concept of potentially editing human cells at the stage of sperm or egg, or most often an embryo. So what if we could change the entire genetic makeup of a person before they're born? So this is called germline editing, because your germline is the cells and DNA that basically are passed along from generation to generation. And here I'm showing you, this is basically like a little thing holding an egg. So this is an oocyte, like a human egg in the middle. And then on the right, there's a needle coming in. And this is how people do artificial insemination or in vitro fertilization. They'll they'll inject sperm directly into an egg through this little needle on the right. And this has been popularized. This happens a lot. A lot of people are born as quote unquote test tube babies or through IVF. But what if we added the CRISPR molecules into that needle along with the sperm? We could just as easily deliver that. And people have. So why is this significant? I think it's important to think about the distinctions between doing a gene editing trial with someone like Victoria Gray, who has sickle cell disease, who is an adult who can consent to a trial like that, and who's only going to be edited in her somatic cells. So somatic cells are non-heritable and only one person is affected. So if Victoria Gray has more children, they're not going to get the same genetic edits that she's gotten. Those will not be passed down. If someone were to edit her germ cells or an embryo produced from her germ cells, that would be heritable. So that kid would be born, they would grow up, and if they had kids, they would pass down the same edits to their kids. So that means that by doing something like this, we're affecting an entire lineage of people, which is much more weighty, when you're making these decisions than just one. And so this introduces so many ethical questions. I can't even get into them all, but I'll kind of go through a few. So there are technical risks with CRISPR, which I think are important to consider in any application, not just germline editing. But in this case, as I said, because this is going to impact the entire body, not just one organ or something that you're trying to treat. It's going to be throughout someone's entire body if they are edited as an embryo. It could have a much worse consequence if something went wrong, right? You would have some kind of bad mutation everywhere if it didn't go correctly. So there's technical risks like that. Most people would say, I would say 99% of scientists would agree that CRISPR is not ready for use in embryos specifically at this point. There's also, of course, philosophical considerations. Various religions and other kind of ways of thinking don't want people to ever mess with the germline. They don't think embryos should be manipulated at all. There's lots of differing views there. And then I think I'm kind of probably most concerned about societal impacts. Like, I think the technical risks can probably be overcome. And once that happens, then it's really more of these philosophical and societal risks that we have to think about. So this could be really expensive. And this applies to any treatment, not just the germline. So it's something we think about at our institute a lot, like how do we make sure that we develop therapies that people can actually afford? Because similar therapies that don't use CRISPR but are kind of the same concept, that have been approved, and the FDA approves it and everyone is then allowed to access it in theory, they have cost like $500,000 to $1 million or more per treatment. And yes, in theory, you would only need a treatment once if you were fixing the underlying cause, but that's still such a huge cost, that we want to make sure everyone is actually able to access this. So that, I think, could be particularly true for germline editing. Like, not everyone can afford to do IVF already. Imagine if you could pay extra and change something about your future child. I think only some people would be able to do that. Only some people would want to do that. But it could create kind of disparities either in health or appearance, aesthetics, if people are trying to use this for non-medical reasons. And then I put the Hulk here. This is very exaggerated, but this is just to imply the concept of enhancement. So like I just kind of alluded to, you could use this to try to prevent a disease, but you could also in theory do this to try to make someone stronger. So we have some knowledge of these genes that control different things like muscle growth or hair and eye color. So you could start trying to design people, which I think is kind of a creepy concept. And I would say for the most part, like nobody would know how to do that right now. We don't have that advanced of an understanding of how genes control traits. So it's kind of not really theoretically possible right now, but there are a handful of genes that we do sort of know about, where someone could start experimenting with this. So if our understanding advances, is that the kind of thing we want people to be able to do? I think the best analogy, the way I try to think about it, is sort of like plastic surgery, right? Like you're just doing that for an aesthetic change, for the most part. But is that something, if you could decide whether or not plastic surgery should exist in the future, if it didn't now, would you say yes or no? Do you think that's overall having a good impact on society? Or do you think maybe everyone should be able to access whatever they want? There's lots of different views on this. And so, yeah, I started sort of alluding to designer babies. And this is something people have been talking about for decades, but now it's more possible than ever. And it actually happened. So 2 babies were born that were edited with CRISPR, and they were edited in theory to prevent them from being able to contract HIV in the future, which I think was a very, very strange choice, because it was definitely not medically necessary to do that. It's just like you could maybe not get this if you're exposed in the future. It's not like you will die when you're 5 years old if you don't get this. So it seemed very unnecessary, and it was actually done very poorly. This guy forged ethics approvals. He totally screwed up the whole science side of it. And so I'm not going to get into that too much. I really recommend this article in The Atlantic by Ed Yong, if you want a nice summary of like every way that this went wrong. So this was terrible. This happened in 2018. And I think it was the only slight silver lining of this was that it was a little bit of a wake-up call to people that we need more oversight of this. And there had been an effort before this from the National Academies of Science, to make this sort of guideline book that says the conclusion of this was don't do this yet. Don't use CRISPR to edit DNA of embryos. But somehow that scientist read this specifically and said, oh, this is great. I'm just preventing HIV. This is within the guidelines. So he thought it was fine. And that kind of spurred people to think, well, maybe we need better guidelines if someone could have interpreted it that way. And so now even more groups are kind of in the mix. So the World Health Organization, for example, has assembled this committee with a very long name, to try to come up with some guidelines for this and to come up with frameworks that could potentially be applied globally, from different countries. So that's another concern, is how do you regulate something at a global level? Because this could be done anywhere. And I should note that right now in the US, it's not legal to do this. And in China, where those babies were born, it was quasi-legal, kind of hard to tell if it was legal or not. The guy ended up being prosecuted criminally, but I would say that's probably because he did so many obviously wrong things, like forging approvals. And this committee, the last time I looked at the website, the only update was that they're in the process of developing a governance framework. So no huge updates from them, but I think that the World Health Organization is kind of poised to do this well, and there's other efforts in the mix as well. So I don't like to end on like, oh my God, designer babies. So I like to point out and bring the conversation back to all the really positive things that we could do with this technology. And I think 95% of scientists in this field are all working on these things. I would say, I could count the number of labs on my hand that are trying to do embryo editing. And even of those, most of them are not trying to do that to make actual babies. They're just trying to study human development. So the things that we can do are really endless. The only limit is human imagination. You can do basic research, you can understand nature, you can start creating clinical therapies for these thousands of genetic disorders that are out there. You can do things in agriculture, like I'm showing chocolate as an example, because we have a project trying to engineer cacao to be resistant to viral and fungal diseases that infect that crop, and can wipe out a farmer's entire livelihood and prevent us potentially from having chocolate. So there's so many things that we can do. It's really a profound step forward in science. So if you want to learn more about this, I really recommend these two resources. So A Crack in Creation is a book or an audio book that you can listen to or read from my boss, Jennifer, and a former grad student of hers, where it's a couple of years old now, so it doesn't talk about the CRISPR babies, but it really goes into the same concepts. And I think it's a great overview of everything. A new book is coming out soon about this as well from Walter Isaacson, who does a lot of like well-known biographies of people. So that should be coming out soon. I think it's called Code Breaker. I can't vouch for it though. I haven't read it yet. And then the other thing is a movie. So if I would watch the movie because I'm lazy, but this is a documentary about CRISPR called Human Nature, that I think was really well done. There's always going to be different ways you can tell these stories, but I think it's cool in this one that they really go through the discovery and show you like who are the people that actually did this work. And you get to see the scientists and see their different personalities and see like, I could be a scientist too. I'm kind of like that person. I'm definitely not like that person. You kind of can imagine like these are real people that do this. It's not people in a movie in white coats that are all walking around like robots. Scientists are people. So I think it's a really cool movie. It's on Netflix now. At least last I checked, it's on there. So hopefully if you want to check it out, you can access it. Okay, so that's it for me. Feel free to follow me. I'm only on Twitter, but IGI, my institute, is on all these platforms that I have here. And we talk all about CRISPR, we talk all about our research, the field in general, different things like that. That's where we would put out different educational resources that we come up with too. And our website has all kinds of information as well. So stay in touch if you're interested. And I think with that, I would be happy to take any questions.
Amazing. Thank you so much. First of all, this is fascinating, a really cool big picture take on CRISPR and all that y'all are doing. So we have a lot of questions, a very engaged audience.
Great.
The first question was about cystic fibrosis, which you mentioned a couple of times at the beginning. Is it possible at this juncture to cure it when it starts? If so, when was that technology available?
Yeah, so there is no CRISPR therapy for cystic fibrosis yet. It's definitely, I think, high on the list in the diseases that are kind of being tackled at the research level at this stage. It takes a really long time to decide I want to cure disease X and then actually develop a whole therapy for it. And so, I know people personally that are working on trying to do that, but they're not at the point where it's actually available. So in theory, you could do that sort of editing at the germline level, like I was mentioning. That would be the easiest, quote unquote, technically to do, to edit an embryo, but that's not going to happen. So this is not going to be happening, I mean, at least not anytime soon, as a way to prevent this before it starts. But it could be applied in children potentially, or in adults, if we get to that point. Right now, I didn't really mention this in the talk, but I think it is kind of important to note that the ability to deliver these CRISPR reagents or CRISPR tools into people's cells is a huge limiting factor in us actually applying this. So with CF, that mostly affects lungs. And we don't have good ways to get CRISPR into lungs. And so even if we could do that editing, we can't actually get those tools to where they need to go. So there's a lot of effort from our institute and others trying to create methods for doing that.
Totally. Thank you. We have several questions on kind of the nature of how CRISPR finds what it's supposed to, how it actually gets there, and how it knows it's in the right place.
Yeah. Yeah. So CRISPR, so the Cas9 protein, that's like the most famous one, that's what I'll refer to. That protein actually searches somehow through all the base pairs in our DNA. This is millions of letters, right? It's actually able to search through all of this and somehow find this single target that is just 20 letters long. That's like the size of that the protein sits on. And that process is kind of understood, kind of not understood in some ways. So we know that it doesn't slide on the DNA. It kind of like bounces across or floats around and kind of checks out some DNA. Is this a match? Is this a match? Is this a match? It might slide a little bit. So there's some things we know about that process, but it's still kind of mind-boggling that it's able to do that. So once it does find a target, assuming somehow maybe you have enough of the protein in a cell that there's lots of the Cas9s floating around, one of them finds the target, it unwinds that DNA, and as long as it has base pairing across the whole RNA, so the entire RNA it's carrying base pairs with the DNA, that's how it knows it's found its target. And I can't even begin to explain. I just was in a talk that was all about like the physics of how that's happening. It's a really long answer, but the protein itself is really cool, and it's just able to sense when that base pairing happens. Like imagine you're zipping up, that's kind of pushing this part of the protein here, that moves this part a little bit, and it just has these little like conformational changes that trigger it to cut. So I can't answer it very quickly, but that's
no, I think that's great. I mean, it's so cool and fascinating to think about the intricacy of that process and the fact that we are still able to target something specific in all of that DNA. So very cool.
Right.
The next question, the video of CRISPR working in real time was very cool. How was that video shot? You said something about nuclear force imaging.
Yeah, it's called atomic force microscopy, AFM. And I barely know anything about this. It's an interesting technique. It definitely has like downsides as well. So we know that in a cell, that DNA, when you saw it cut, it kind of like flew away, right? In the cell, it actually stays where it is. So Cas9 stays bound over that cut because it's bound so tightly, and it has to get knocked away in order to actually have the editing happen. So we know that that thing doesn't actually happen the way that it was shown in that clip. So that I think is like a notable downside. Like this is not necessarily recreating the exact physics and everything that's happening in real life, but it's super cool. I mean, I was like blown away. Normally I'm holding a tube of clear liquid and like imagining what's inside it all day. So to actually see this thing was really awesome. So as long as you take in those caveats, you can really learn a lot. So I can't begin to explain atomic force microscopy, but you can look it up.
Absolutely. I mean, my mind was blown watching it. So very cool.
Great.
I think our kiddos are very inspired by you. Had one question about if a kid is in high school right now and they want to get to where you're at, what's some big pieces of advice, some big next steps that you would have for them?
Sure. I guess I would say first, like, don't try to get where I'm at. Follow whatever curiosity you develop. Like, if you think, okay, science communication sounds really cool, I'm going to pursue that. But then at some point, you take a class in like history that you like better. Like, feel free to switch. I think people definitely like have passions, but don't be afraid to take opportunities that arise and kind of go with the flow. Like, I know everyone that's in my career line got here in a weird way. So they were just doing something else and they kind of discovered that this existed, and no two science communication jobs are really the same. So I guess I would say just always try to follow what you're interested in. Definitely parts of school can be really boring. Like you have to go through some of it. But when you actually have the choice and you can pick a class that you get to take, definitely just try to follow your interests. And when you get to college, and I don't know, maybe even now, if you're near a university, there are so many free talks and seminars happening all the time, and movie screenings, panel discussions, things like that. And so when I was in college, I really tried to go to lots of things. So it was really fun to see. I would go to like some cognitive development lecture about babies and how they learn to recognize their parent or things like that, which of course it's like 90% over my head, but it's just kind of cool to get to see a little bit. So don't be intimidated when things are confusing. Like the more you learn about stuff, the more you realize you will never understand most of the world. But just as long as you're following your curiosity and you're happy about what you're doing, I think you're on the right track.
Cool. I think that's great advice in general. I think very few of us know exactly where we're going to end up, especially within the fields of STEM, but continuing to pursue what you're passionate about is really good advice. Okay, next question. You talked about editing physical traits like the Hulk and making designer babies, for example. Do you think it would be possible to edit the physical traits of an adult?
Yeah, so that's a tough one. I mean, I think it depends on the trait. So some things are kind of locked in. Like, I don't know that you could change the eye color of an adult. I would have to know more, I guess, about how eye color happens. But I think sometimes once you've finished developing some trait, it's there forever. So it might not be, you might not be able to switch something like that. I think, I mean, with hair, maybe hair is different because the cells are there, but the hair itself, like you can shave it off, it can fall out. You're making new hair all the time. So I think it'll depend on the trait. And that's a question that applies also to like disease. So there's some diseases like Marfan syndrome, for example. It's like you end up growing in a really elongated way. A lot of the tissues and muscles are weaker because they're so stretched out. And so for something like that, I'm not sure if a CRISPR-based therapy would be enough at that point once all these structures and things are developed, but it could help to some extent. So again, it's going to depend on the trait in most cases.
Okay, awesome. Thank you. One question about basically how you think in the next 2 decades this technology will evolve in just in terms of accessibility, where you think it will be.
Yeah, let's see, next 2 decades. So CRISPR has only been around for like 8 years at this point, and it's already in clinical trials. It's already potentially cured that one person I showed you of a disease. So that's actually very fast for something like this to go from like just something someone found in a lab to in people's bodies. That's really fast. So it's a quick pace, but it is just so slow. Like you might have a false impression of how clinical trials work because of the COVID vaccine, because it's all on like hyperdrive. Usually these will take years. You have like phase 1, phase 2, phase 3, and you have to go through just really slow processes of testing these things because it's not guaranteed that these things are going to be safe in every instance. So we really don't want to bring them out before they're ready. And you have to do that for every single condition. So it is kind of a slow process. I think I would hope in 10 to 20 years, we would definitely within 10 years, I would say we will have FDA approved CRISPR therapies for maybe a handful of conditions, assuming things continue to go well. In 20 years, I would hope there's dozens and dozens. It is a hard problem. Like I was mentioning, a lot of rare diseases have like 5 people in the whole world that have one. And in that case, you're not going to go through this whole clinical trials process. So we've been talking about, and I think lots of people from FDA are talking about like, how do we do these really individualized treatments for people and scale something like that? Because you don't want to just abandon people because they're too complicated. And there's not money to be made with a blockbuster drug that millions of people are going to take. So I hope that we get there. I hope we kind of figure out some innovative ways to approach that problem.
Yeah, cool. I mean, really exciting to think about the future of where this could all lead, and especially in terms of like equity and accessibility for lots of populations where there isn't money to be made. So I think that's important. 2 more questions for you, and then you are off the hook. The first one is about germ cell mutation. So when we edit a cell in the germ cell, but it's mutating and passing that along, how does that look? How could that affect the lineage line, etc.?
Yeah. So if we were to edit the germline, hopefully you would just make the edit you were trying to make, if someone's even allowed to do this at all. Hopefully you would make the edit to, prevent a disease or something like that. And the reason that gets propagated, I guess, or spread to all of the cells in the body is that when you're editing a single cell, it then divides into 2 cells. those both have the edit, those both divide into 2 cells, all 4 have the edit. So that's why it's able to go everywhere. You would hope that there's not mutation happening. That can happen just during normal development with nothing to do with CRISPR. If you do get a mutation when the cell's dividing, it's copying the DNA each time it divides, it makes an error. That can cause cancer. There can be a lot of different things that can happen if that happens to you. So I guess you would hope that CRISPR is not causing additional mutations, but if they were, those would also potentially get passed to all those cells. So yeah, I'm not sure if that answers the question, but hopefully.
I think it does. I mean, again, I'm reading the question, so I think it does, but I mean, that kiddo may not. Last question, which is about you. What was the most fulfilling thing that you've done in your career so far?
Fulfilling. Oh, that's hard. I don't know. I've enjoyed a lot of it. I guess I really like when my friends ask me science questions. So during COVID, that has happened a lot where I get to I feel like I'm actually helping someone directly, whereas a lot of scientific research, you're so far removed from the people that would actually benefit. It's kind of cool to be able to tell your friend, get the vaccine or wear a mask or these sorts of things, or more complicated questions. So I'm kind of most fulfilled, honestly, by my personal connections when I'm able to use my science background to help them, or even if it's not about COVID, it could be about anything. But I don't know, I've really enjoyed a lot of it. It's a fun career path, and you get to do just so many different kinds of things. It's really cool.
Okay, awesome. Well, thank you so much. I know that this is super meaningful to our students and teachers. They have been super interactive during this, and just your expertise and also just honesty as to where we're at in this field right now is much appreciated. So thank you so much.
Great. Thank you. Thanks for having me.